Good morning, everyone. It's Mark Crowther, Chair of the Department of Medicine here at McMaster University, and I'm joined by two guests today, Dr. Zane Chagla and Dr. Shelley McNeil. I think we all know Zane, having watched him on both our own little computer screens as well as on TV and lots of other places. He's become a real media personality. I noticed that the lighting in his Zoom thing has improved dramatically since the start of COVID, and also his background and the tidiness has improved as well. So he's becoming a professional not only at COVID, but also at Zoom presentations. And also Dr. Shelley McNeil, who's the director of the Canadian Centre for Vaccinology based in Halifax, Nova Scotia, and is the vice chair of the Department of Medicine in Halifax. And so Zane's going to talk a little bit on diagnostics, IPAC, and treatment, and Dr. McNeil is then going to talk about Canadian vaccine developments and where we are with the vaccine. And I think that is of excruciatingly high priority for many people on this webinar. I've managed to turn off the chat function this week, so I won't be haranguing you about not putting things in the chat function because you physically can't. If you have questions, please put them in the question and answer box. I will monitor the question and answer box. Those questions which can be answered in real time, I'll attempt to do that. Other ones, I'll collate them and present them at the end. It's very unlikely that we'll have time for all of the questions. We have never had time for all the questions, so I apologize in advance if we don't get to your questions. These are being recorded and will be placed on the Department of Medicine YouTube channel for subsequent viewing if you wish to watch them later. So given we've got a lot to talk about, there's been lots of changes since the last time we did one of these updates a few months ago. I'll just move right directly to Dr. Chagla. So Zane, please go ahead.
Hi, good morning, everyone. I'm happy to be here, and we'll just do a quick update in diagnostics, IPAC, and treatment. I think obviously a lot of us are very curious about what happens in the second half of this talk in terms of vaccine development, and so we'll hopefully allot more time for that and then keep questions at the end. So the major updates in diagnostics have been the release of more rapid-based assays in Canada. There are two assays that are on the Canadian market that the Canadian government is currently trying to distribute in multiple provinces. There are other platforms, rapid PCR platforms, that are also approved but have not been picked up as much. The two major ones that you've been kind of headlining the news are the Abbott ID Now. This is as Donald Trump is caressing the ID Now here, the methodology that was used in the Rose Garden affair, which unfortunately does put a bit of a damper on some of its effectiveness. This is essentially an isothermal molecular reaction in a box, so it is not perfect PCR, but pretty close. So it is a molecular test. The patient has a swab that's inserted. It goes into the sample receiver, which has the liquid media, and then the test space goes in. It's closed. You run it over 15 minutes. This thing's, as you can see from it being held, about the size of an espresso machine, and you get an output in 15 to 20 minutes. Its sensitivity is about 83 to 92%. This is in symptomatic individuals. Specificity of 94 to 100%. Turnaround time in about 15 minutes. The major Achilles heel with this is that it is a single test per analyzer. So it is hard to run these in sequence unless you have multiple analyzers all at once. You really have to go through the actions, insert the cartridge, shut the cartridge, wait for a result, dispose of the cartridge, and then restart again afterwards. There are apparently validation issues if this device is actually moved to, so it has to be recalibrated. So again, it is more friendly for a stable setting like a lab in a long-term care facility. And so we are looking at this device currently, predominantly looking at it for a couple of models. One is as a model for remote settings as a first test, along with a lab-based PCR method. So in some of our smaller rural hospitals where that may be a difficult go. And number two, an interesting look at whether or not it could be an outbreak in a box mechanism. So as we know of our issues in long-term care, being able to deploy one of these as part of a rapid testing strategy as people are developing febrile illnesses or a staff member is tested positive as part of surveillance testing, really starting to look at that first ring of very highly risked contacts and staff, trying to figure out from there who's kind of clean and dirty, who needs to isolate, who doesn't need to isolate, and then moving on from there along with lab-based testing. So there is a lot of potential here, certainly for this device, but it is not as easy as kind of putting it in a doctor's office and having a rapid result. I think you still have a lot of lab accreditation to deal with it, but certainly a lot of potential for more of a sensitive test in terms of patient care. The second, and these are part of the sequence of rapid antigen tests that have hit the market. So this is the PanBio, also by Abbott. It is a lateral flow assay and essentially a nasopharyngeal swab. And from what I understand, in the coming quarter, nasal swabs have been sent to Health Canada for approval too. The swab is taken, it's put it into these tubes, which are actually quite cool in the sense that the swab breaks off and the tube actually turns into a dropper as well. So you don't necessarily have to pipette out of the tube. It turns into its own pipette. It's put on the lateral, the buffer's added in, it's put on the lateral flow assay, and essentially you get a control line that should light up positive and a test line that lights up positive in the context of a positive test. These have a sensitivity of about 70 to 75% and a specificity of about 95%. The sensitivity issue is quite interesting, and realistically, it is partly operator error, which might deal with some of the sensitivity. But there is also the concept of a viral load that is relevant, and a lot of discussion with this in Michael Mina's lab in the United States. Where these tests perform well, the viral loads tend to be high. And as we know with PCR, we do get people that shed positive for some time. So, whether or not that sensitivity is actually clinically relevant sensitivity versus gold standard sensitivity is one of these concepts that's really being flushed out. The other part is, again, these are probably not great to pick up people very early in their infectious process, but done repeatedly could theoretically increase that sensitivity. And so these have been approved for symptomatic patients with COVID-19. This is still a clinician-derived test that the clinician still needs to do the nasopharyngeal swab. But the government is really trying to roll these out in a different way. And so we're looking at use cases actually where that sensitivity can be combined with a relatively low pretest probability to then really have a relatively good negative predictive value. And so we're talking about settings like long-term care screening, where this may be a whole lot easier to do and may not be as much of a burden on the provincial testing supply as a method to actually preserve that. There's lots of other use cases that are being trialed around the province, and these have been distributed to us at St. Joe's amongst a number of other sites. They've been distributed to long-term care facilities. They're even going to workplaces to see if they validate in those environments. And I think, globally, these POC tests have been used as the standard of care where PCR test is prohibitive, hard to license, or delays are huge. And I think, again, better the laboratory and low pretest probability populations. If you, are on Twitter, want to follow Dr. Michael Mina, he has a really, this concept of testing everyone repeatedly with these antigen-based tests, repeatedly with less samples, compromising some of that sensitivity and saying, hey, is this a method to get back into society? Is this a method to just, continually test people to isolate the really, really infectious? And probably there are some population effects from it, and not to worry about the sensitivity issues amongst these tests if you're using them for that methodology. I know Shelley's on the phone and her colleague, Dr. Lisa Barrett from Dow as well, had looked at this amongst a cluster in Nova Scotia, and particularly in Halifax, where there's been some spread of community transmitted disease, and then just inviting people in some of the hot spots to get tested. And I believe a handful of people that were completely asymptomatic, but potentially relatively high viral loads tested positive in that sense. So a good proof of principle there. And I think a lot of us have heard about the Slovakia example. So Slovakia was dealing with a huge burden of disease and really did have to do something very drastically. They then decided to test everyone in 2 sequences. And if you weren't tested, you'd have to isolate for 14 days regardless. So they essentially went out to their entire population. And interestingly, You can see the gray here is test number one. The gray here is test number, gray here is test number one. This was a test. Gray here was test number one. The gray here was test number two. And you see, a rapid drop in their average cases, particularly positive PCR tests, as you essentially isolated people from phase 1 and 2 in their contacts. You also see a very real corresponding drop in the reproductive value. And again, as we talk about better markers, as hospitalizations are probably much more of an objective marker, you are seeing a stabilization of hospitalizations, which is what would be expected in this context, and not necessarily the archive disease that people are getting, or would still result in hospitalization, but certainly a stabilization. And so, some really interesting thoughts about this strategy is a mass testing. I think this is data that is hopefully going to be published soon, but does give that credence to say, okay, a huge mass testing effort, even with a lower sensitivity test, similar to what they're doing as well in Nova Scotia, may take some of these infectious people out of the population, especially when burdens are high. It's not perfect, but again, it's the enemy of perfection is good, and it might be just enough to get things under control. Moving on to infection control, I think one of the big things was this update in terms of aerosol transmission. And realistically, this is something that the World Health Organization has talked about, the CDC has talked about. I think we have a paradigm in IPAC about the large and small droplets. Whereas with many infections, I think we're realizing that there is a spectrum here, where in circumstances where people are more crowded, in circumstances where ventilation is poor, that there is a risk of more aerosol-based transmission, meaning that things can generally spread a bit further than the 2 meters and can spread in very higher inoculums. There's the classic choir example. But locally, I think we learned Spinco may have been an example of some of this too, where a significant number of people, despite having the 2 meter spreads, where masks were down, people were breathing heavily, and the ventilation may not have been optimal, led to a significant number of infections. Is this the bottom line around this? I think we know this is a possibility, and definitely is something that should be investigated more as we're talking about community-based transmission and how to optimize that in many settings. In inpatient care, particularly in hospitals, I think we follow ventilation standards that are incredibly specific. And I think that does negate a significant amount of this risk having air exchanges, which is probably not as applicable. And for now, even the Public Health Agency of Canada, Public Health Ontario, multiple other provincial agencies, the World Health Organization, even the CDC, have really not changed the precautions as related to this as part of COVID care for patients in inpatient settings. I will put a plug out though for all of you that are doing COVID care in Emerge on COVID units, where there is still equipoise amongst this. Dr. Loeb is still running the N95 versus surgical mask study. And for those of you who are interested, there's information on both of the wards and in the hospitals, and you can feel free to contact me or Dr. Loeb if you are interested in registering, as I think it's a critical study to finally answer the last bit of this question. There are some updates on masks and really what kind of masks are appropriate. And so this really does go based on a couple of things. One is the fact that outpatient or community-based masking has had a number of fairly ineffective masks that are used. The neck gaiter, this mingle mask thing, which is essentially a face shield on your lips, and other slightly crazier examples. There's a great 22 minutes bit on this too for people who want to take a look. Realistically, and looking at some of the aerosol scientist studies, particularly one by Lindsay Marr that's in a preprint now, that looked at filtration capacity, not only from a source control standpoint, i.e. the mask protecting me from contaminating the environment, but also from a personal protective affair, that really the 3-level masks were better than 2, and the addition of a middle layer, such as what's in a surgical mask material, or even the vacuum bag filter, would be relevant for outpatient and community-based use. The masks we use in the hospital, again, meet these standards to a significant degree, But again, thinking about the masks that we use in the community, I think we've all seen examples of poorly fitting masks and masks that really are made of very flimsy material. And I think this just sets a standard for what should be there for people to use for their own personal protection as well as source control. The one study I will bring up, and there are some issues with the study, but I do give it to these Danish authors to actually try to study the use of masks. So this was a study that came out a couple of weeks ago in the Annals of Internal Medicine. And this one was done in Denmark during their real decline in COVID, so April to June, where masks were not recommended at that point. They recruited people who had 3 hours outside of the house. They were randomized to getting mailed surgical masks versus not masking, and had antibody and self-directed PCR tests. And they really tried to power for a 50% reduction in COVID. They weren't able to achieve with that power just because this was a guess based on this being a relatively new intervention, just based on other studies. And so there were a lot of drop-offs obviously here, given that it's a relatively difficult study to control, especially in the context of COVID-19. So not being able to kind of physically see people and get things done. But, at least 2,000 people in each arm from 17,000 recruited, which is still not bad considering, the context of doing clinical trials at this time. These people had antibody and oropharyngeal, nasopharyngeal swabs, nasal swabs, that were self-administered. They did antibodies pre and post. They did swabs monthly, and then they did swabs at the time of symptoms, as well as healthcare swabs if they sought care. And so this was the primary endpoint. There were 42 versus 53 people, and no difference in the odds between those who developed COVID-19 in either group. And in antibody plus PCR, again, no difference amongst them. It's interesting that many of them did develop it based on PCR, either in healthcare or asymptomatic PCR testing, whereas many developed antibody positive diagnoses rather than healthcare associated diagnoses, which also is nice fodder to say that probably we aren't diagnosing as many as should. I mean, the Canadian Blood Services data suggests maybe 6 times more are antibody-based diagnoses than swab-based diagnoses. There are a lot of limitations to this study. So this was really based on personal protection. So you're wearing the mask, how much COVID do you get? It's really a great example of source control. So what effects are in society when people have their droplets blocked? The powering may not have enough, and I think they overestimated the size of effect. There's obviously the healthy volunteer effect. So people that are probably willing to do this study, that are probably willing to also adhere to public health precautions. There was minimal community transmission. If you look at the use case, there was actually quite a bit of poor use of the masks. Although one would argue, knowing what we see now, people do wear their masks, but there is some degree of poor use or partial use there. So, that might actually more mimic the general population. But the bottom line, I think, is masks on a population level are unclear effects from a personal protection standpoint, probably from a source control standpoint. I think many studies do suggest this from modeling. There may be some degree of personal protection, but again, the hierarchy of needs puts PPE at the bottom, and I think that really does reflect that, from a personal protection standpoint, things like distancing, not going out while sick, are much more effective than true masking. I just want to quickly go over some of the stuff around de-escalation of precautions. Early in the pandemic, we were swabbing people for negativity. This was probably not a great idea, given that this is a viral infection. We were looking for molecular signals. And so we really, from other bacterial and viral infections, we often find them after people are resolved, and the clinical implications are fairly minimal. There's multiple studies that have been done in the interim looking at CT values, and Mushevik here in the Lancet has really done a nice meta-analysis of them. Really, none of these studies show viability after day 9, even with people that have relatively high viral loads, which is reassuring. In our jurisdiction, again, we deflag most at day 10 if they febrile and improving, and critically ill and immunocompromised day 20. There are a few cases that are emerging in the literature now of people who are CAR-T or stem cell transplants that have had prolonged viral shedding of 40 to 60 days based on viral culture. And so, this day 20, particularly for the immunocompromised, I think people could still consider using the viral swab in that context, in the severe stem cell immunocompromised issue. But generally, I think we're getting much more understanding that our cycle thresholds probably even don't correlate particularly well at that point, and that day 10 is probably very safe for most people in that sense. And then we were talking here about the quarantine rules, and I think there's been a lot of press about this. This was yesterday. So the CDC is set to actually shorten the length of quarantine to day 10. So this is exposed individuals, not people that are positive. So day 10 rather than day 14 if you don't get a test, and day 7 if you get a test, recognizing that most people are positive relatively early, as well as most people will show up positive by day 10. Is there going to be a risk here? Absolutely. There's going to be the odd person that shows up day 11, day 12 with symptoms. It's very unlikely. And again, the whole point here is if you would rather have more people that are acutely infectious isolating or quarantining from all of us, making it much more palatable if you can deal with 7 days, rather than necessarily trying to treat the 1% that might stay out for day 14 in that sense. And again, rational work. There's a lot of discussions ongoing locally whether or not this will be applied. Lastly, moving to therapeutics, I just wanted to cover a few drugs. I think dexamethasone, the recovery trial, has been relatively well uptaken and has become the standard of care, with a number needed to treat of 6 or 7 in the ICU and a number needed to treat of 25 outside. I really do give it to the recovery authors to be doing a trial like this in the context of everything, and really ongoing trials that really try to prioritize all patients with COVID-19 into a clinical trial. This is the remdesivir data, and then again, a bit of controversy here. This is the final Act 2 trial that was published in early November. This is 1,062 patients, 500 that were in drug and placebo. They did see the median time to recovery was shorter in the group that got remdesivir, and clinical improvement by day 15 was better. There was no difference in mortality. And looking at the subgroup analysis, the category I do want to take it to is much of this was fueled in these mild patients. The ordinal scores really deal with 7 being ECMO ICU, 6 being high flow BiPAP, and then 5-4 being mild, and 5 being oxygenated. You see the mortality really that moderately ill oxygenated patient really driven here in the hazard ratio, and then the ordinal scale recovery again really driven here in that group. And so this was the recommended group potentially after ACT to really consider for the use of remdesivir, whereas the critically ill patients and the mildly ill patients probably would not derive a benefit. This is balanced by the Solidarity trial, which we have been contributing through to CATCO, where essentially 2,700 patients who got remdesivir versus 2,700 that got standard of care, which was open label, that saw no difference in mortality, as you can see in the Kaplan-Meier curve. No difference actually in lopinavir, interferon, and hydroxychloroquine, which are interesting compounds that had largely been discounted based on their data, but really no difference here in a large trial in terms of mortality, ventilation, and discharge. And interestingly, in that trial, they did do a forest plot of all of these studies done to date. So Solidarity here, where there might have been a signal in the low flow group for mortality, but again, by no means significant. There was a small Wuhan trial that also looked at this, and then Act 1. And so the point estimate here, when you go through that lower risk, no ventilation, might favor this data from Solidarity, this data from Act 1, where the mild group might derive a benefit here. But arguing to that is the only thing that is actually fueling that is the ACT data, and whether or not that's relevant or is that an anomaly. And so these are the recommendations, and Brahm and his group with the World Health Organization, the BMJ, had really put out these rapid recommendations. And really, remdesivir is not recommended for. Corticosteroids are recommended week not recommended, sorry, recommended against in non-severe and recommended for obviously in severe and critical. And the NIH recommendations do put remdesivir here in the severe requiring oxygen. The Ontario Clinical Practice Consensus Criteria said could consider, but prioritize clinical trials here. And I think locally, you know, there is a small supply that has been provided by the government. I think for open label, obviously, we want to give it here. And for those places that are offering clinical trials, prioritizing clinical trials first, because I think this group needs to be flushed out a lot more, and it's not going to happen without clinical trials. The last slide I'll mention here is a monoclonal antibody trial. The only reason I mention this drug is approved in Canada, and we have bought $27 million worth of dose. And this is the interim analysis, which really funded the approval. So this was a 3-to-1. They used 3 different doses versus controls. These were mild, not hospitalized individuals with COVID-19. The thought is this drug may not be a good drug for, you know, people that have made it to hospital. Their inflammatory storm is fairly high. This is for the high-risk individual in the community that then needs, you know, something to prevent them from getting into hospital. Recognizing logistically that's fairly hard with an intravenous compound in someone's home or in some sort of outpatient setting. Primary outcome here was viral load. Secondary outcome here was hospitalization. Viral load is interesting, as you can see here, that the dose that actually achieved a viral load response was actually the middle dose, not the smallest dose and not the largest dose, which always puts things biologically into some context, saying, well, if we gave more, why didn't it do less? And then secondary outcomes, similarly for viral load changes, were really seen in that middle dose more than anything else, which again is a bit fishy in terms of how this study is actually fueled in that sense. When they looked at secondary rates, there was 1.6% hospitalization in the antibody group, 6.3 hospitalizations in placebo. But if you look closely, the N is 5 versus 9. They thought this was significant, obviously, given the placebo group is 143 and the COVID monoclonal antibody was 309. But I have a lot of issues with this trial leading to an authorization is fine, but leading to a wide scale distribution and $27 million worth of drug being bought in Canada for relative data that doesn't make a lot of biologic sense. It might decrease hospitalizations, but it's still not clear yet. I think there was a lot of time to go, as this was an interim analysis, to say whether or not this was cost effective and useful. And with that, I will switch it over to Dr. McNeil here to go through vaccines for the next little bit.
Thanks, Zane. Let me just share my screen here. Are you seeing that okay?
Yes, yep. We can see it fine, thanks.
Excellent. So thanks so much for having me. A pleasure to virtually see all of you. I did put a disclosure slide in here. I'm a vaccine researcher, so have relationships of one sort or another with everyone who makes vaccines, essentially. My only learning objective really was to review what we know about vaccines for COVID and what might be soon to come in terms of how we roll them out in Canada. So as you may be aware, there are over 200 vaccines in the pipeline for COVID-19. However, there are all of them fall into 6 different vaccine platforms in terms of technology and mechanism of action. So the first ones we're going to start seeing very soon are the Pfizer and Moderna vaccines, which are mRNA vaccines. I'll talk about them in more detail. Followed quickly thereafter, hopefully, by the AstraZeneca vaccine, which is sometimes called the Oxford vaccine, being developed in collaboration with Oxford University. And then shortly after that, again, hopefully subunit vaccines, and then the rest will follow much later. Canada has pre-purchase agreements with 7 of these products, all falling within the early group of products expected to be available. And all of which are starting to report phase 3 clinical trials data, at least in the form of press releases. So the New York Times, as you probably have seen, has a nice tracker for vaccines. And as I said, in mid-November, there were 212 candidate vaccines in development, 48 of which are in clinical trials and 11 in phase 3. There are none authorized for use in Canada or the US, but as you may recall, Russia licensed a non-replicating viral vector vaccine a ways back, and hot off the press yesterday, the UK authorized the use of the BioNTech Pfizer mRNA vaccine, and is actually expected to begin dosing next week. So that should be very interesting and informative to watch. Just a teeny bit about the virus itself and thinking about what we're choosing as the antigen. So the gray surface here is a spherical envelope that contains the genetic material of the virus. The little orange bits are membrane proteins, or M proteins, and they're the most abundant structural protein and give it its shape. These do vary virus to virus, so it can help to identify one virus from another molecularly or otherwise. The little yellow bits are envelope proteins, which play an important role in replication of the virus, assembly and release of the virus. And finally, the red clumps are the spike proteins, or S proteins. And it's these spike proteins that allow the virus to attach to cells and enter the cell, fuse with the human cell and enter the virus to release their genetic material. The spike protein is the dominant virulence factor for the virus, and it is actually the antigen that's been selected to be the primary antigen target for all of the vaccines we're about to talk about. So here you see again the pathogenesis of the virus. So here you see the initial entry of the virus into cells, primarily type 2 pneumocytes, after binding to the functional receptor, which is an angiotensin 2 receptor on the surface. After endocytosis of the viral complex, the surface ACE2 is downregulated, and that actually results in unopposed angiotensin 2 accumulation and local activation of renin-angiotensin-aldosterone system. And this is felt to mediate lung injury responses to the virus in patients, and often resulting in more severe lung disease. So the first set of vaccines I wanted to talk about are the mRNA vaccines. The Moderna product and the BioNTech Pfizer vaccines that have recently reported phase 3 clinical trial data publicly, efficacy data publicly, are both mRNA vaccines, and they will be the first 2 vaccines used in Canada. Messenger, or mRNA, that codes for the SARS-CoV-2 spike protein is enclosed in a lipid envelope or lipid nanoparticle and injected as the vaccine. The lipid allows the mRNA to get into the cytosol of the cell, and it uses the cell's own ribosomes there to translate mRNA into the spike protein, or the antigen. The antigen then can take 3 paths. It can be processed in the cell and presented directly on the cell surface, where it activates B cells, stimulating a humoral immune response. Or it can be processed by processed and presented on MHC1 on the cell surface, stimulating a CD8 T cell response. Or finally, it can be endocytosed, transported to the lymph nodes, presented on MHC class 2, where it stimulates a CD4 T cell response. So there are actually 2 nucleic acid-based vaccine platforms, the mRNA vaccines we're talking about now, as well as DNA vaccines that are further behind. mRNA is less stable than DNA, and that will be relevant when we talk about the limitations of this vaccine with respect to storage and transport and moving it around. The mRNA is very unstable, and Pfizer actually uses a term jiggle factor because they don't actually think that the vaccine is particularly stable once it's not frozen. So once this vaccine is thawed, it can't actually be moved. So it will be, when we move forward, it will be difficult because patients or people who are being vaccinated are probably going to have to come to the vaccine rather than sending the vaccine out to the people who need it, which is going to be a challenge. RNA does have the advantage, though, of only needing to take one step. It enters the cell, it's translated directly, it only needs to cross one membrane, doesn't need to get into the nucleus. And of course, there's no risk for mRNA to be integrated into the genome of the cell. DNA has this extra step of needing to get into the nucleus where it's transcribed to mRNA and then translated to produce the spike protein. And there's a hypothetical, at least, or yeah, I suppose mostly hypothetical risk that the DNA could be integrated into the host genome. And the concern there would be that you'd get abnormal translation and potentially the development of something like malignancy. So the advantages of the mRNA vaccines then are that they can be produced very quickly. You can interchange the antigen easily if need be. And they have been shown, as I said, to induce both cellular and humoral immune responses. The big disadvantage is this is the first time we've used mRNA vaccines in humans. They are known to be poorly stable. And of course, both companies are working on ways to make the vaccine more stable. We don't have any data about the durability of these immune responses. And of course, there will be need to monitor long-term safety. So the two, again, are Moderna in partnership with the NIH in the United States, which is a non-replicating mRNA lipid nanoparticle, exactly like I showed you in the first graphic. And it has, again, just reported phase 3 results. And then the partnership between BioNTech in Germany and Pfizer, very, very similar vaccine, also finishing phase 3 trials in the US and Germany and some new phase 1/2s in Japan. And both of these companies have advanced purchase agreements with Canada, so we'll be providing vaccine to Canada as soon as the vaccine is authorized. This is the summary of the late stage results from these two vaccines. Again, this is science by press release, which we're all in the business of these days. Pfizer, I'll talk about first, since it's the one that we likely will see in the very near future, did have the advantage in the clinical trials, all done in the US, of including children and a significant population over the age of 65. Was tested in about 43,000 people. It is a 2-dose vaccine, as are most of these, given 2 weeks, in this case, 3 weeks apart, some are 4. This analysis was the final analysis done of the phase 3 efficacy trial. It was triggered when 170 cases occurred in the trial. 162 happened in the placebo group and 8 in the vaccine group. The data were collected 1 week after dose 2. Really remarkable efficacy demonstrated of 95% for the prevention of infection. And even in the over 65 group, they had sufficient power to report efficacy of 94%. The vaccine also appeared to reduce the risk of severe disease. There were 10 cases of severe disease in the trial, 9 of which occurred in the placebo group. Moderna also recently reported its phase 3 results in adults in the United States. They tested their vaccine in 30,000 US citizens, again, 2 doses, 4 weeks apart. This was an interim analysis triggered at when 95 cases occurred. 90 occurred in the placebo arm and 5 in the vaccine arm, again, a couple weeks after the second dose. Again, remarkable efficacy. These are very, very similar vaccines, so one would hope that they would perform similarly, and they did. So 94.5% efficacy, but notably no reports yet about subpopulations, particularly older adults, which of course is important. Also appeared to reduce severe disease. There are 11 cases of severe disease in this trial, all in the placebo group. No serious safety concerns identified for either vaccine, but important to remember that we only have median follow-up of just over 2 months in the participants in these trials who received these vaccines. So it will be important to have ongoing safety surveillance. And both vaccines are quite reactogenic, which will be important when we're administering them to manage expectations and not immunize, for example, whole floors of healthcare workers who then might all stay home the next day because they're feeling crappy. So this is the Pfizer mRNA vaccine. The final dose selection was 30 micrograms, which is these middle bars. The first chart here is local injection site reactions. You can see that virtually everyone, I guess everyone, had pain at the injection site, mild or moderate pain, not much difference between the first and the second dose. Also some redness at the injection site and some swelling. But importantly, again, you're looking at the middle bar in these clusters of 3. There were quite a few systemic adverse events, notably fatigue, headache, chills, muscle pain were quite common after this vaccine, and about half of people ended up taking some sort of medication to manage side effects. So again, will be very important to manage expectations. This is the Moderna vaccine. Again, final dose selection was 100 micrograms, so kind of the middle bar. Local injection site reactions are on the bottom. Again, you can see that most people had pain and some redness at the injection site. Systemic adverse events were also common. About 70 or 80% had at least one systemic side effect. Interestingly, with this vaccine, it's nicely visible here. You can see that after the second dose, the reactogenicity was much greater than after the first dose. And that wasn't as clear with the Pfizer product. So again, important in terms of managing expectations. So then I'll turn to viral vector vaccines. The third vaccine that's recently publicly disclosed its phase 3 efficacy results was AstraZeneca in partnership with Oxford, so the so-called Oxford vaccine. This is a totally different vaccine than the two mRNA vaccines we were just talking about. The Oxford vaccine is an inactivated viral vector vaccine. So viral vector vaccines use a non-virulent or attenuated virus to express the antigens of whatever you're trying to induce immunity against. In the case of the AstraZeneca or Oxford vaccine, they use adenovirus as a vector, and you take essentially viral vector vaccines are produced by taking a piece of the SARS-CoV-2 RNA that codes for the spike protein, using reverse transcriptase to create DNA, to generate DNA, and then that DNA is integrated into the host viral genome of the attenuated adenovirus. So these viruses can be attenuated, replicating viruses, in which case the virus itself turns out SARS-CoV-2 spike protein as it replicates. Or as is the case with the AstraZeneca vaccine, you can inactivate the viral genes that enable adenovirus replication and then still infect the cell with, and still provide the cell with the ability to make spike protein. So the advantages of viral vector vaccines is that they do clearly establish both humoral and cellular responses without the need for an adjuvant. They can potentially boost immunity to the vector virus. So adenovirus, for example, causing common cold, there could be some benefit to its use in a vaccine. We do have several vaccines, 3 in fact, vaccines in the world using this technology. 2 are Ebola vaccines using a vesicular stomatitis virus vector, and then there's a dengue yellow fever vaccine made in this way. The potential disadvantages of the vaccine really relate to the immune responses that may exist to the vector virus. So if seropositivity against the vector virus is high in the community, that may reduce immune responses. And that, of course, might be different by age. So children who may not have seen the vector virus may respond better than older adults who have had more exposure to the vector virus. You may also potentially see blunting of the response after the second dose for the same reason, with antibody development after the first dose. And if this becomes a technology that we use in the future, any future vaccine that uses the same platform could obviously also be impacted in terms of its immunogenicity. So there are lots of these viral vector-based vaccines around and being developed. The 2 that are furthest along in development are the Oxford vaccine and a Janssen and Johnson & Johnson vaccine. The AstraZeneca vaccine gets around the issue of immunity in the population to the vector by using a chimpanzee-derived adenovirus viral vector, and it's in phase 3 trials in many countries around the world. The Johnson & Johnson vaccine uses adenovirus 26 and is currently in phase 3 trials in the US. So here are some of the results that we have available. Not much yet from Janssen, but AstraZeneca again has released publicly some phase 3 data. These trials were done in the United States with an adult population, again 2 doses. The primary efficacy endpoint, they reported an efficacy of 70.4% overall. But this one you probably heard about in the news. There was a dosing error in the cohort, the first part of the cohort, under 55-year-olds who received the vaccine early on. And in that cohort, the first dose was miscalculated and they received half the planned dose in the first for the first dose, and then the error was corrected and they received the full dose in the second for the second dose. And interestingly, the efficacy in that population was 90%. If in the group that got half, then a full dose. So moving forward, they will be using half, then full dose. I don't really understand why that happened. There's been lots of smarter than me immunologists speculating about it, but it is what it is, I suppose. So a fortuitous error. They've not reported anything about response to the adenovirus vector. You'll recall that there was 2 pauses in this trial for neurologic adverse events, but both were investigated by an independent data safety and monitoring board, and the study was reinstituted. So we don't have any more detail about those. Janssen & Janssen, just some phase 1 data showing brisk humoral and cell-mediated responses and no real problems with reactogenicity. The AstraZeneca Oxford vaccine, though, is also more reactogenic than the average adult vaccine. So in the bottom panels here, this is the control vaccine, which was a quadrivalent meningococcal vaccine. Up here is the AstraZeneca vaccine. The with and without prophylactic paracetamol or Tylenol. Not much difference between the two, but you can see again, injection site tenderness and pain were common after this vaccine, and lots of systemic adverse events. Again, notably fatigue, headache, joint pain, myalgias, chills. So all 3 of these vaccines, which we're about to start using broadly, do have a, fairly unpleasant reactogenicity profile. So again, we'll have to manage people's expectations so they know that this is expected and not to be worried. The last group is the subunit protein-based vaccines. Shortly behind, this would be the Medicago vaccines, Novavax, and a partnership between Sanofi and GSK. Subunit vaccines are produced by identifying the portion of a pathogen to which you want to generate an immune response and creating that protein recombinantly, in this case, the spike protein of SARS-CoV-2, and in heterologous cells like yeast cells or other insect cells. In the case of the COVID vaccines, of course, the spike protein is produced recombinantly and either packaged with an adjuvant. In the Novavax, it's a matrix M nanoparticle adjuvant, and in the GSK Sanofi trial, it's a proprietary GSK adjuvant. And then the spike proteins form a nanoparticle, which is the antigen to which the immune system is exposed. Or the recombination to form the spike protein can be altered at the end to allow the spike protein to self-assemble into virus-like particles. These have good immunogenicity because the immune system sees what appears to be the whole virus, but of course they can't replicate because they don't contain any genetic material inside. So these, we do have experience with subunit protein-based vaccines, both nanoparticle vaccines and VLP vaccines, including Hep B and HPV, which are virus-like particles, and other vaccines that are nanoparticle vaccines, like some meningococcal vaccines and Haemophilus influenza vaccines. So again, these are the 3 companies, Medicago, Novavax, and Sanofi, making these vaccines. The first 2, Medicago and Novavax. Medicago is a Canadian company making VLP-derived, plant-derived VLP vaccines, just starting phase 3 trials in Canada. Novavax is being done in the UK in phase 3, and a little further behind is the Sanofi product, so there hasn't been any data, as far as I'm aware, released about that one. And again, all 3 of these companies have advanced purchase agreements with the Government of Canada. The data available for these is really just early phase data showing really impressive humoral antibody responses. So responses that far exceed that seen in convalescent sera following acute infection. but also nice cellular immune responses and no untoward reactogenicity. So it is interesting that it's a novel technology that we've never used before that will be the two vaccines that we start with, but keeping in mind that we have a really robust regulatory process in place to look at the safety and efficacy of these vaccines before licensure. Again, we'll see the Pfizer and Moderna vaccines come out quickly, and they do induce protection very soon after the second dose. But remember that we don't know a correlate of protection. We don't know the duration of protection that these vaccines will confer. So really an impressive timeline, less than a year really to first availability of vaccine. We would have expected it to take, upwards of 15, 16 years if we did our normal, plodding through the vaccine development as we would normally do, with each thing happening sequentially rather than concurrently. But using some really tremendously interesting and innovative adaptive trial designs, as well as funding that took away risk for companies, this process has been dramatically accelerated such that all of these things were happening concurrently, including down to building buildings and preparing to manufacture and ship vaccine. Pfizer actually has vaccine shipped into the United States already, anticipating licensure on December 10th. Oops, sorry. So behind the scenes, of course, there's lots of work happening in Canada to assist with the vaccine rollout. There's a COVID-19 vaccine task force chaired by Joanne Langley here in Halifax, and Mark Lavonin, who used to be the president of Sanofi. And they've been helping the government really looking at this early data, largely to help decide which vaccines the government should be investing in, to ensure that it has vaccine available. They've also been working with companies around all of the bits of the filing that need to happen in order to secure an early and appropriately quick emergency authorization. Health Canada has agreed to a 15-day review. They've been meeting back and forth with the companies all along. They're doing rolling submissions. So typically they would be waiting for all of the data to be available in a package at the end, but they've been accepting data as it becomes available and reviewing it on an ongoing basis. Again, so that when the final data is provided, they can provide an approval very quickly within 15 days of the last data coming into them. And again, we have pre-purchase agreements. You'll note that we only need about 75 million doses to vaccinate everyone in Canada with 2 doses. We have purchased 194 million doses with option for another 220. And that's largely in case one or more of the vaccines either don't work, aren't safe, or we can't actually get them in Canada. In terms of implementation, there will of course need to be guidance on who gets the vaccine first. And that guidance does come from the National Advisory Committee on Immunization. They have released some preliminary thoughts on that issue, with key populations identified for early vaccine use, including those at high risk of severe illness and death, largely resting around age, but other as yet to be defined high risk conditions. Those most likely to transmit to those people. So that would include healthcare workers, personal care workers, and caregivers in long-term care facilities, those that are required to maintain other essential services, and those in living conditions that would put them at high risk. So congregate living settings, First Nations, corrections, shelters, that sort of thing. They have tried to refine that guidance a little bit because recognizing now that there may actually be vaccine coming out really in the next 1 to 2 months, we do need some more specific direction to think about. So the plan, like in the UK, is to start with people over the age of 80 and expand down by 5-year increments as vaccine becomes available, and to target residents of long-term care, assisted living facilities, and other older residential settings for older people. Then concurrently with that, healthcare workers working in long-term care facilities will be a first priority. They don't actually mention caregivers in the most recent statement, but I suspect that was the intention. And then other healthcare workers considering exposure risk. So that may include ICU, COVID units, those sorts of things. And then finally, they have recommended targeting rural and remote Indigenous communities in the first go around. So some specific challenges, these are difficult vaccines to administer. The Pfizer vaccine stored at -70 needs to be transported on dry ice. And as I mentioned, once it's thawed, it can't be moved. Each tray in the shipping containers contains 1,000 doses. Once you reconstitute it, it has to be used within 6 hours. And then we have to give 2 doses to everyone. So it truly is going to be a miracle if we can pull this off, honestly. Moderna, similar problems, although not needing the low temperature freezers. And then once AstraZeneca's vaccine becomes available, it's more of a normal sort of ship for cold chain vaccine. And I'll not say much about this in the interest of time, but of course there are plans in place for post-marketing safety, adverse event surveillance, through many different mechanisms in collaboration with the provinces and FAC and some research surveillance networks, as well as plans to look at real world effectiveness moving forward in Canada, particularly against severe outcomes. And we all have to try to debunk the misinformation available out there. People have equated the rapidity of vaccine development with sloppiness, which of course is not the case, or I hope I'm speaking to the converted, but is not the case. Only 69%, 63 to 69% of Canadians said they would get a vaccine when they were asked in October. And 40% of them said they would get it, but they wanted to wait and see how it went with other people before they gave it. So we could have a problem if we can't get good information out and make sure people get vaccinated. So with that, I'll stop just by saying, it's been remarkable, the process to get to licensed vaccines, but now we really have to focus on implementation and the science of implementation and social science to ensure the people who need the vaccine can get it quickly. So I will stop there. Thank you very much.
Thanks very much, Dr. McNeil. And we've got a number of questions here, and Zane too. I'm going to cherry pick a couple of questions that weren't covered during the talks. I'm going to ask Zane this first of all, and then Dr. McNeil, maybe you could comment. Dr. Mullner's asked, what will the policy be with respect to healthcare workers that refuse vaccine? And I presume there is no such policy, but I'll be interested to hear what you think.
Yeah, I mean, it's a good question. I think we're wrangling with this now. It's, obviously we're weighing the risks and benefits here, right? So we have to see what the data says. We have to see what the safety data says. If there's not a medical exemption there, then I think given the predisposition for hospitals to have outbreaks, there may be more of a strict policy in mind here. But again, that's based on efficacy, that's based on safety, that's based on a number of things we just don't have access to right now. And Shelley probably can deal with these discussions more ethically in that sense.
Yeah, I mean, having fought the mandatory influenza vaccine and healthcare workers battle for the last 20 years, I would be very surprised if we could mandate this vaccine. So, we would obviously be thinking about whether people who can't or won't be vaccinated should be working in certain areas, for example. But I can't imagine that we would make people receive the vaccine if they don't want to. We just might have to think about how better to protect them and patients if they're unvaccinated.
Thanks very much. There's a couple of different questions that are all about high-risk populations, and I know there's no data in high-risk populations because the studies haven't been done. Are you, Dr. McNeil, are you aware of any plans to do studies in other populations, for example, children or pregnancy or immunocompromised people that specifically target those people?
So I know that vaguely in the wind, I know there are plans to do that. Pfizer has been enrolling children in their trials. There are some other pediatric trials starting up. Zane probably has more information about those. But so I think the answer will be yes, but I think they're just trying to get through these primary efficacy pivotal trials first. And then, like with many vaccines, we'll start to see target population trials coming soon.
A couple of questions. If a person has been documented to have COVID before, any information on whether they should get vaccinated, whether vaccine would make any difference for them? We probably don't have any data, but a couple of people have asked that question.
Yeah, the 2 mRNA vaccines had subgroup analyses where they included people with evidence of prior infection, both of which showed still similar antibody responses. So I don't know that all trials, many of them have excluded people who had evidence of infection before. So I don't think I have enough information to answer that question. except that I think there is a subgroup analysis in either the Pfizer or Moderna that did include some people who had prior infection with no untoward outcomes.
And the other part I'll mention is, if you were talking about a mass vaccine campaign, we're not going to be doing antibody testing to say, did you have COVID or not, right? It's going to be, as we know from the seroprevalence data, there's a lot of people that get COVID that don't get a test, that don't know they had COVID. So at the end of the day, people are going to get the vaccine that have had COVID in the past, like it or not.
Yeah.
And you'd presume that in the U.S. studies, been a lot of people who've had COVID who, if they aren't doing serology beforehand, if they do serology, we know it's not very, there are areas where it's not reliable. So there will have been a number of people who would have had COVID and then got vaccinated subsequently and never realized they had COVID. And if it had some kind of weird toxicity, one presumes you'd start to see that cropping up because there'd be a high rate of weird toxicities.
Yeah, I'm sure that will be one of the subgroup analyses. I mean, we need more information, obviously, in all the subgroups in these trials to know how well the vaccines work in different populations. And that would be one population they hopefully will have data on.
So we're a bit over time. I'll ask one last question, and then we'll just end. And I apologize to those who didn't get their questions asked. If someone wanted to participate in a clinical trial of vaccines that's currently underway, are there any running right now? How would one actually access those trials? Do either of you have any information on that?
So there definitely are trials going on in Canada. I know the Canadian Centre for Vaccinology is involved in several. So clinicaltrials.gov is probably the easiest place to go look because it would list all of the vaccines. You can search it by Canada, would show you all the locations in Canada participating in vaccine trials.
And Zane, are you aware of anything going on locally?
I am not, actually, not for this one. There's other vaccine trials for other diseases ongoing locally if people are interested, but yeah, I don't know of anyone that's even through C.
I think
Allison
McGear's doing at least one or two of the trials that we're doing at CCFB. So okay. I think there might be some, but I don't know the specifics. Nearby you, I know you're not right there, but okay, Ontario's, Ontario, just like we're the Atlantic bubble now.
I'd like to, we should probably end because it is past 9. So I'd like to thank Dr. McNeil for traveling virtually from the Atlantic bubble to present today. Thanks for a great presentation. She's the director of the Canadian Centre for Vaccinology and really an expert in this area. And of course, our own local celebrity, Zane Chagla, who's been doing an awful lot of great work in this domain. Thanks, Zane, again for appearing back in this. We are planning on doing another update. We've currently got it scheduled for early March. In the chair's grand round. So, obviously we'll have one sooner if there's some breaking developments. If not, we'll get a comprehensive update done in March of next year. I'd like to thank everybody for watching. Wish everyone a unusual holiday season because this is going to be an unusual holiday season. My plug for something entertaining in a tough time is if you go to YouTube and you type in Ryan Reynolds and Taylor Swift, you'll see a great 2020 ad that they put together that was released yesterday that I found personally very hilarious. And so I would strongly recommend you go have a look at that if you want something a little bit humorous today. And I wish everyone the best. Look forward to getting my vaccine when I'm in the queue, whenever that is. And thanks again to our presenters and to our attendees. And we'll all talk to you all later. Thanks very much.
Thanks very much.
Bye-bye.